Happy Monday evening. So it's the week 14 on this newly outlined course. And we are looking at the Monday evening's review session for the exam 3 and practical 3. So what I'm going to do, I'm going to just jump right to it and we'll work through the material for this session. So as we are getting started, I just wanted to acknowledge there has been few messages that I'm just about to answer after this session, asking for extensions and due dates to be moved ahead in terms of the previous work. So I'm going to do that. I want to make sure that you have a chance to practice the material before taking the exam on the chapter content. And for this week's session, we have 4 of these, obviously. And this session, I want to focus mainly on going through the study guide for the exam 3. There are, as always, a couple of other things that I will also mention and cover with you. One thing that I wanted to kind of celebrate or share with you before we get any further is kind of having a look of the newly adjusted schedule. Obviously, as you can see, after the week 8, we had the spring break, and then that was extended by another week for the week 10. And now we are at the week 14. So we're actually about 3/4 done with this course. So we are on the winning side of the project. So I really want you to give yourself a big pat on the back and remember that there's only a little ways to go, and make sure that you get all of those points towards your final grade that are available and up for grabs. In terms of this week's coursework, there is no new assignments that need to be done per se. Instead, I'm asking you to complete the exam 3 and lab practical 3. Both of those are due at the end of this week on Sunday, which I believe is the 25th. And make sure that you notice that there's quite a bit of points up for grabs on both of those. So I really encourage you to tackle both. And like I said, the biggest focus for this week's sessions are to go through the study guide for the exam 3. And as always, I have made the study guide available for you in a version where there are little gaps here and there. And the on time that we get to really fill in those gaps are as part of these exam review sessions, which I'm organizing 4 of those this week. And of course, these are also made available to you as a recording. So what we are going to do at this point is actually to jump out from this presentation. And instead, I'm going to pull up our study guide and we will work through that together. So let's get that up and running. Here we go. You should be able to see the study guide for the exam 3. And let me just make it a little bigger so we have a little more to work with. And as always, on this session, it is my goal to go through the main points that are relevant to our chapter content. We're covering the chapter on the respiratory system, the chapter on urinary system, and then the final chapter, which is kind of a continuation of the urinary system, looking at the water electrolyte and pH balance. So as always, all of the questions in the exam are pulled from this study guide. So it is a good idea to make sure that you get a chance to have a look of that. And we are going to make a start by jumping right into covering the respiratory system. So that's going to be our focus. And I think that that's one of the larger chapters where we go into a greater detail. And as always, I want to kind of put things on a map for us to start with. So we are starting by looking a little bit of systems and how they are connected to each other, and especially in terms of the respiratory system. So, of course, looking back, what we have discussed as a previous parts of this course, we talked about the cardiovascular system. So, cardiovascular system works to assist respiratory system in transporting many of the respiratory gases, nutrients, waste products, hormones, and so on. Really, if I would have to pick couple of most important ones, of course, the transportation of oxygen and carbon dioxide become very important for the respiratory systems side of the view of looking at what the cardiovascular system does. We have also talked about the lymphatic and immune systems, which serve a role in the body defenses. So really their function is to protect us against pathogens. And that applies also to, in case of the respiratory system. So if you think of the respiratory system, it is an open route for a large part coming into our body. So we need to make sure that those pathogens, foreign invaders, are not able to get and cause damage inside our body despite it being open. So that's where the lymphatic immune system plays especially important role in our discussion. And then let's wrap up these connections with other systems by remembering the urinary system. And main function of the urinary system was, of course, the regulation of the blood pressure. But in addition to that, urinary system does a lot of other things. And one of those in the angle or in the point of view from our discussion on the respiratory system is that it helps us to deal with those hydrogen ions that we might want to get rid of. And we'll talk about the bicarb and transportation of that a little bit as part of this course. So now that we have had a look of the systems connections, I think it's a good place for us to kind of consider the importance of the respiratory system. And you might remember that I told you that there's three main points that we worry about when we first approach a patient, whether it's in an emergency medicine, whether it's as a paramedic, or whether it's in the first aid setting. We're checking for three things, and two out of those three things are actually dedicated for the respiratory system. So we're checking for the airways. Is the airway clear? Is there a clear path for the air to enter to the lungs? And we check, is the breathing happening? If there is an issue with the blockage of the airway, we try to clean that. And if there is no breathing happening, we might end up having to do those rescue breaths, giving mouth-to-mouth resuscitation. And then the third one, which is not related directly to the respiratory system, is the compressions, chest compressions. If the heart is not pumping and moving, the role of that is to make sure that we generate at least some amount of artificial movement to the cardiovascular system to transport oxygen, especially to the tissues, so they don't get stopped. And the organ that we're especially worried is the brain. So this brings up back to this familiar formula that we might have seen, which is all about the cellular respiration. So we see here we have sugars, which as a part of the process of cellular respiration are turned into ATP, which was of course the energy currency of the of our cells. But to do this, we do need oxygen. So we need to take in oxygen to the body, and we need to transport that to those cells where the cellular respiration happens. And as a byproduct, we are left with carbon dioxide. So we need to get rid of that carbon dioxide somehow. And same goes for the water that's produced as a byproduct. But from our perspective, really, we are especially interested on this: how to get oxygen to the tissues and how to get carbon dioxide away from those. So this is really putting everything on a bigger map, kind of reminding ourselves why the respiratory system is as important as it is. So we are seeing the main functions, again, just highlighted there. Getting the oxygen into the body, so supplying oxygen to the tissues, and removing the carbon dioxide from the body. There are other functions that the respiratory system serves as well, such as in the sense of smell. The proper term for that is, of course, olfaction, and production of the speech as well, which is super important for modern humans as a communication form. There are other non-respiratory movements that our respiratory system participates as well, and we'll have a look at those in a little bit. How we are going to cover this chapter on the respiratory system? So we're going to look at the different parts of the respiratory system, going through them one by one. And in addition to that, we are then going to move on to have a look of the physiology. So this chapter was really divided kind of onto two parts, more of an anatomy heavy and then more of a physiology heavy. But as always, you remember that there's no anatomy without physiology, so there's overlap between the two. And the first that we're going to start with is going to be, of course, our nose. So we're going to start by talking about nose, why nose is important, what role a nose plays in the respiratory system. That's going to be our first topic of interest on this review. And as we remember, most of the air is taken in through the nose, and we also get rid of most of the air through the nose. So breathing through the nose is the normal way. When additional intake or output of the air is needed, then we do have an option of recruiting the mouth, but that's not our first option. We first choose to do the breathing through the nose, and we'll have a look, and it will become much clearer to you as part of this review session why the nose is the ideal solution. There is times when we do recruit the mouth, as I said, and one of those times, an example would be yawning. So what happens in yawning? Often I hear the psychological explanation, more of a social explanation, that it's a response to being bored. But really what yawning from a physiological sense is to recruit the mouth and oral cavity for quickly adding in more oxygen when our body is giving us as indication that oxygen levels are kind of low. So that's what yawning serves mainly, and that's why it's easy to see the saying that yawning spreads throughout the room. Well, if the oxygen levels in the room are low, then it's more likely that more people will have that physiological response to that. Well, when we start having a look of the nose, one thing that we notice is that they're at the nostrils, nares for singular and nares for plural. Within the nostril, we do have the hair. And let's do a quick sketch here. Here we have the nose. And you'll notice that we have the nasal cavity separated from oral cavity with our palate, and we have these hair right at the first part of the nostril, and their function, like we already concluded, was to filter air. So that's kind of a coarse filtering. Any large particles get removed there. We make sure that we get the biggest parts wrapped already out of entering to the body at that stage. The next part that we're going to look at is going to be the nasal cavity. And like we saw on that diagram that I just drew, nasal cavity is located immediately behind the nose and nostrils, so our focus will be on this part. And what we notice is that the nasal cavity is really lined by a mucous membrane. And this is an important term. And mucous membrane is this kind of a thinner than skin, very sticky, humid surface. And the stickiness of the mucous membrane is generated by the mucus that's produced by the goblet cells. And the function of this mucus is to trap any particles that we might not want to have entering to the body. And hold on a second, my cat is playing with something that she shouldn't. Can you leave those feathers alone? Excuse me. Well, I'm getting silent treatment when she got caught. Let's continue. So mucus and mucous membrane really is the second way of filtering the air, making sure that nothing that shouldn't get doesn't get in. And let me just pause for a second. I'm gonna have this thing addressed with my cat. Okay, my apologies for that. Anyone who has pets in the house know that the time when you need to focus on something else is the time they would decide to get involved with things that they shouldn't. But let's continue looking at our nasal cavity. And I'm going to sketch another diagram here. This diagram is going to be a cut of the face showing the nasal cavities. So we would have our eye sockets here from the front. The nasal cavity is divided into two by nasal septum. And then for the nasal cavity, we've got these protrusions, three of them coming out from the wall of the nasal cavity. And these protrusions have a particular name and a role in the process as well. These are known as the conchae. Conchae are simply protrusions from the lateral wall of the nasal cavity. Let's add them to our diagram here. And they serve actually a twofold purpose for us in sense of the respiratory system. So, of course, we have our most superior, middle, and inferior ones of those. First of all, they increase the surface area. Then the importance of increasing the surface area comes from the fact if we think what was the function of the nasal cavity, the mucous membrane there, it is to catch as much of that air filtered as possible. And we'll discuss about a few other functions as well. And the other thing that these conchae do, they generate turbulence, if you wish to use that term. So you can think of that as a horizontal kind of a twister or horizontal airflow, making sure that as much of the air as possible is circulated within the nasal cavity and gets in contact with this mucous membrane that we had a look. I already mentioned earlier on our nasal septum. Well, nasal septum was the part of the nasal cavity that separated the left and right side from each other. So what we saw there. And finally, the nasal cavity is separated from the oral cavity by the palate. And let's sketch another quick diagram here. See, there we have the eye socket, the nose, the mouth of an individual. And let's sketch the nasal cavity there and the oral cavity as such. And what we see here is that the floor of the nasal cavity, if you wish, is made by the palate. And for the palate, we have two parts. More anteriorly, the hard palate, And it's called hard palate because there is actually bony structure within here. And then more posteriorly, we have the soft palate. And soft palate term comes from the fact that there we do not have the bony structure inside there. It's just soft tissue, and all the way at the back, at the most posterior part, we of course have our uvula, the hanging protrusion there at the very back that you can even see from the mouth. So that's our nasal cavity briefly described. Other thing that we notice when we discuss this region of structures is the paranasal sinuses. And let's do a couple of sketches to discuss about the paranasal sinuses. And as you remember from our earlier discussions, a sinus is always some kind of an cavity within an organ or within a tissue. So paranasal sinuses refer to these cavities, empty spaces within this area. So again, just to draw quickly, our diagram of our skull of the head here looking from the front. So we have our teeth and then the mandible going up there, and rest of the skull if you wish. That's the front view. Let's add to the bottom also a side view. So here we have the eye socket, the nasal cavity or nose, and then we have our mouth here with the teeth on the upper jaw and lower jaw, our mandible coming up, and rest of the skull there. So we're going to go through these paranasal sinuses, these openings within the skull on the region or on the bones of the skull. So there's going to be 4 groups or 4 types that I really want to focus on. And we're going to start with the frontal sinus. What you will notice that all of these sinuses are named based on the bones where they are located. So the frontal sinus would be sinuses on the left and right side would be located within the frontal bones. If you want to kind of visualize them roughly on a sketch, they're of course above the eye sockets, these little empty spaces there. Let's add it also to the side view of the skull there. And the next one that we are having a look of is going to be our maxillary sinus. And again, one on the left and one on the right side on the maxilla bone. So right underneath the eye sockets, these are probably the larger sinuses that we find within the body of the paranasal sinuses. And that's from the front view, and on the side view we can add it there as well. Now, the next one that I want to discuss with you is going to be the sphenoid sinus located within the sphenoid bone. So that's going to be a little harder to illustrate on the view looking straight of the skull. We're going to illustrate it instead on the side view. Remember, it would be within the bony structures within the skull, within the sphenoid bone. So it's not lateral, that it's more medial. And the final structure that I'm going to add is going to be the ethmoid sinuses. So there's multiple of those. Another term, a term that I prefer to use more of to describe them is the ethmoid labyrinth. And the reason why I prefer to use that is that the term labyrinth really describes their presence and their kind of connection. They're all slightly interconnected sinuses, small here behind the nasal cavity and within, or behind the nose and within the nose region of the bones within the ethmoid bone. Well, there's naturally the next question that I would ask if I was a student, and I can think of you asking from me, is that why do we have these sinuses? Why do we have these empty spaces within the skull? What purpose could that possibly serve? And on our material, I think that we covered 4. So we're going to focus on those. Some of them better accepted, some of them a little less accepted. But regardless, we're going to go through all of them. Probably the 2 first ones are the most well accepted within the field. That first of all, these empty spaces within the skull help to reduce the weight of the skull. So if you think of the bird and bird bones, those are hollow. That makes the skeleton of the bird a little lighter, easier to fly, easier to lift that weight. So the same idea has been proposed for the purposes of the paranasal sinuses of the skull, that if it was a solid bone, it would be much heavier, much more work for us to carry with our neck. So having these sinuses would serve a purpose of reducing the weight, at least to a certain extent. Another thing that we do notice with sinuses is that they play an important role in the voice resonance of an individual, in formulating how your voice actually sounds, how your speech sounds. So they're important in there. The third one is a little bit more debated one, more of a theory at this stage. It has been theorized that just as when you have a car, and I'm going to draw a really simplified diagram of a car here. There we have the headlight with the lamp shining. A couple of windows there, and let's add the tires as well. So anyone who has driven a car and had an unfortunate situation that they might have ended dealing with a car accident, and if it has been a head-on-head collision or head collision, what you'll notice is that the front of the car has been actually designed to collapse in an accident. And the idea behind that is that that absorbs some of that impact, so that the force that results from the car stopping as it crashes head-on first or something is not going to be as great. So some of that force is absorbed there, so that the impact on the passenger, on the driver, is not as strong. And it has been speculated that the same could be possible for the reasons of the paranasal sinuses of an individual, that they also would kind of collapse if there is a direct impact with the skull facing head first to some sort of a accident, some sort of a rapid stop. So the fourth function for the paranasal sinuses, which seems to be well supported, is that we do see that they are connected to the immune system, so they serve a purpose on that sense as well. And now taking a step a little bit on the side of the clinical aspects as it relates to the paranasal sinuses, we have most heard about the sinusitis. You remember itis was always an inflammation of something. So in this case, it's going to be an inflammation of the sinuses. So that can happen as well. Another structure that we do end up finding within the nasal cavity would be the opening of the auditory meatus, so the connection to the inner ear from the nasal cavity. That's there if you, for example, go to a higher elevation, maybe on a plane or driving up, or even have a pressure change as if driving through a tunnel. You can hear your ears sometimes getting clogged, and the way to deal with that is by swallowing, so that stabilizes that pressure difference between outside and inner ear. So that's why we have that opening there. Well, now we get to what I promised to you, that I will tell you what is the main function, if you wish so, of the nasal cavity at large. And there's 3 functions that I really want us to note for the nasal cavity. And remember especially that mucous membrane lining, because it plays an important role in facilitating these three functions for us. So first of all, nasal cavity and the mucous membrane there, as well as the hair at the nares, serve to filter the air that enters into the respiratory system. Another function that we notice is that with the mucous membrane being a thin layer, a lot of blood supply underneath there. So that's why it's easy to get nosebleeds, just puncturing that, damaging that mucous membrane a little bit. It's much more sensitive than skin, for example, would be. But by having a lot of blood supply there, we're bringing in a lot of heat with the blood, and we're able to heat up the air before it enters further down to the respiratory tract. In addition, you remember that mucus was sticky, so we are bringing also humidity there, and that gets picked up by the air before it moves further down to the respiratory tract. So those are the three important functions of the nasal cavity. And if you have ever had to see when this doesn't work, they make sense, especially the number 2 and 3. If you have had a deep breath of cold air on a dry winter's day, you'll notice that that cold air makes you cough. And that's because it's not heated enough and it's not dry enough. So taking a deep breath through the mouth, for example, on a cold winter's day, might cause issues with those. But let's continue our journey looking at the different parts of the respiratory tract. And we're going to move our attention to look at the pharynx. And pharynx can be described as being kind of a muscular skeletal muscle tube structure. It runs all the way from the back of the nose to the start of the larynx. That's about at the level of cervical vertebra number 6. What's noteworthy with the pharynx is that it can be divided in 3 parts. So again, let's do a quick sketch of structures that we already are familiar with. The eye socket there, the nose there, mouth, our nasal cavity taking shape there nicely, the floor of the nasal cavity, the palate, and then let's draw the oral cavity for the sake of consistency there. And here we get to the neck. We can see the Adam's apple, for example, there. So, larynx, pharynx, let's start with the pharynx. So pharynx can be divided in 3 parts, and we're going to go through those parts. And let's start with the nasopharynx. So nasopharynx, as the name suggests, is located immediately behind the nasal cavity. And then we are going to have our oropharynx. Well, oropharynx is located behind the oral cavity. So let's add that to our diagram. And finally, the most inferior part that we see, that is gonna be our laryngopharynx. And let's add that to the diagram as well. That continues to the level of epiglottis, which is one of the structures that we'll be talking in a little bit, and continues in general until the larynx. So that's why the laryngopharynx term to describe that. So those 3 portions for us to know in regards to the pharynx. Let's continue our journey. And next we are going to be looking at the larynx. So more inferior part. Larynx runs between the vertebrae, cervical 3 to cervical 6, if you want to kind of map it out somewhere. And really, if you want to have a landmark from a bony structure that you might be familiar with, one of the structures that you might have seen is the hyoid bone. And hyoid bone is located most superiorly to the larynx, which we will discuss next. So if you're kind of refreshing your memory of the hyoid bone, that was the bone that was the floating free within the neck, not really attached to any other bones, just held in place by muscles, soft tissues, and so on. That was the bone here, easily broken if one gets strangled. So that's sometimes used as an indication in forensic research, but that's not a definite clue. You can get strangled without the hyoid bone getting broken, or hyoid bone can break even if you haven't gotten strangled. But what larynx is made? Larynx is made of a number of cartilages, and in addition of a combination of muscles and elastic tissues. We will, for our purposes, focus on cartilages that are the most important ones as we are wanting to review the parts of the larynx. And the cartilages that we will look at, let's start with the first one. The first one that we will look at is going to be our thyroid cartilage. There's one thyroid cartilage in a larynx. And that's probably the largest cartilage part that we see there. Has a little bit of a kind of a heart-shaped top part, superior part. And we have this protrusion on there that comes out and gives the rise to what we know as Adam's apple. The proper term would be laryngeal prominence. That is the kind of a prominent protrusion that we mainly see mostly on men. Females do have that, but not quite as prominent as what we may see on the men. If you're trying to think of other landmarks or other reasons why you might be familiar with the thyroid cartilage, well, of course, the thyroid gland would be located at this side, at this level as well. And we remember thyroid gland had the two butterfly-shaped lobes and the isthmus connecting those. Let's continue our journey looking at the cartilages that make up the larynx. And the next one that I want to have a look of is going to be the cricoid cartilage. This is the most inferior part of the larynx, and actually the only fully enclosed ring-like structure of this structure as a whole. If we take a side view of what we're seeing, that might become useful a little bit later on as we cover more of these. So I'm just going to add our thyroid cartilage here and our cricoid here. So this is the front, and this is going to be side view. So let's move on to discuss a structure known as epiglottis. This is a cartilage flap-like structure that really serves the purpose of sealing off the trachea whenever one would swallow. So it prevents us from having food or water or liquids entering within into the windpipe. So it's this kind of a flap-like structure inside our thyroid cartilage, if you wish. At all other times, other than when swallowing, it is going to be open. So that tells us again about the importance of securing the airway, that's always open and available for the air to flow through. The next structures that I want to add and discuss with you a little bit about are going to be our arytenoids. There's going to be two of those. So different from what we've seen, there's actually two of those. Again, they are located inside the thyroid cartilage. And they are, if you wish to describe them, kind of a triangular or a pyramid-shaped structures. And there's a rich supply of muscles that's connected to those. And this helps us to manipulate the angle and also, if you wish, the move these arytenoids as necessary. And why this is going to be important, let's add our muscle highlight here. Why this is going to be important is that it is the vocal cords that are attached to our arytenoids. And of course, we manipulate the tension of the vocal cords as we manipulate the location of the arytenoid. So that allows us to tighten or loosen these vocal cords depending on what sort of a sound we're wishing to do. Let's add corniculate cartilages. These are tiny tips on top of the arytenoids. So two of those as well. Let's add them to our diagram here. One on each side. And finally, I want to add also another double structure on one left and right side called cuneiform cartilages. These are actually harder ones to see because they are located within the mucous membrane lining of the thyroid cartilage, but they're located inside there. So those are the main parts of our larynx and what we need to know there. Then, of course, from the bottom of the larynx, we have our trachea, so our windpipe starting from there, and we'll discuss the structure of the trachea, including certain adaptations that we see there with the other cartilages structures, these bands in a little bit as we move further on our discussion. But I'm just adding that there as a sake of completedness, kind of, and help us to navigate of what we're seeing. Well, we should also discuss the function. Why do we have a larynx? Why is larynx an important structure to have? Well, first of all, of course, we saw that that is the airway passage route for the air. It also is the site where the air going to our trachea and food going through our esophagus are separated. Probably another really important function that I want to highlight here is arising from the fact that this is where the vocal cords are housed. So for the production of our voice. And let's add a little bit of detail what we can know about the vocal cords. And we're going to do another simple diagram, kind of map a couple of things that we're familiar with and a couple of new things. So I'm going to draw looking from the top. This is going to be the anterior part. This is going to be the posterior part. Here we have our thyroid cartilage enclosing everything. We have our arytenoids. And from those, let's color those in. So our thyroid cartilage, that kind of houses and closes inside all these structures, are arytenoids, as I mentioned them. And from those were moved with muscles. that help to move the arytenoids. And from the arytenoids, we had our vocal cords. What's interesting and worthwhile to note, there's actually two sets of paired vocal cords. And these are made of elastic fiber. Their white color is an indication that there is not really heck of a lot of blood supply to this region, so there's not much of blood flowing to this area. But the space that's located between the vocal cords has a specific name, specific term that we want to know. That is known as remaglottis or sometimes glottis in a simpler version. I mentioned that there's two sets of vocal cords, more superiorly, something that we call vestibular folds. So vestibular folds are often referred as the false vocal cords, and that's because they don't take part on the voice production, whereas the inferior pair, known as vocal ligaments, are the true vocal cords. They are the ones that play the major role in the production of our voice. Okay, let's continue our journey. And next we are going to discuss and have a look of the structure of the trachea. So trachea is sometimes, on a layman's terms, if you wish, described a little bit more often as our windpipe, if you wish. And it's about a 4 inches long tube that runs from the bottom of our larynx, from our cricoid cartilage, which we saw here, and continues until it divides into two primary bronchi. So again, let's do a quick sketch. So here I'm gonna draw the cricoid cartilage and our thyroid cartilage. And from there, from the cricoid cartilage, we had our trachea starting. And one thing that was really important or very obvious to note here was these C-shaped rings that we have for the entire distance of the trachea. These C-shaped rings, and they're C-shaped if you look from the top down, they have an important function, important role, and we'll discuss why they are C-shaped. So let's draw a quick diagram. And again, I'm simplifying things. This is looking down on an individual and not to the scale, but this individual, the anterior side, the front is here on the left, posterior side at the back. And let's draw one of those C-shaped rings of the trachea. So you might wonder, first of all, why do we have these rings? Well, that's to make sure that the airway remains open at all times. So even if you would go unconscious, the muscles would relax at this area, the airway would still remain open. So that's the important function of these tracheal cartilage rings. And I'm just gonna, for the sake of simplicity, label there the trachea. That's what we're looking there. More anteriorly. Posterior to the trachea, we end up finding our oesophagus. And oesophagus is squashed closed, if you wish, whenever it's not in use. So that's how we don't get burps and stuff. We keep it nice and closed when it's not needed. It's located posterior to the trachea. But you might wonder, why is there an opening at the back of the trachea then? Sometimes you might swallow food that's extremely large and we need to have space for the esophagus to open up. So that would be the purpose of this opening at the back of the trachea so that it can extend there as needed for a larger piece to pass down. Now that we get to the end of our trachea, one thing that we'll notice there is that it divides into two branches. These branches are known as primary bronchi, and we'll notice that the right-hand side, right primary bronchi, I don't know where we jumped there. The right primary bronchi comes down in a steeper angle than the left one. And this point where they divide into two at the end of the trachea, this point is known as carina. That might be an important landmark. For example, if you put a camera down someone's throat, that would be a kind of a good point to be familiar. It does really look like this triangular or a pyramid top. And I mentioned that the primary bronchi, from the primary bronchi, the right one came down on a steeper angle. Well, that explains why, especially when children, babies, infants have a tendency of putting everything into their mouth and sometimes swallowing stuff that they shouldn't. If this object ends up going to the lungs, it is to the right lung where it often ends up going, and it is because of this steeper angle of the right primary bronchi. One clinical example, clinical term that I've included there, the tracheotomy. If we would have a situation where the patient has an obstruction on their airway. So let's think of the obstruction, for example, being here quite high up on the airway. And we're not able to get the air through there, we might need to make an incision here somewhere along the trachea to let air in and out through that opening, and that way make sure that the air supply remains to the lungs and away from the lungs. Next, let's have a look of further structures coming from the trachea. As we move down the respiratory tree, we had our, I didn't mean to use that color. We had our primary bronchi. You remembered separating into the right and left side, with the right being in a steeper angle. And then we have our secondary bronchi going on to the tertiary bronchi. Bronchioles are smaller versions of these. Eventually we have terminal bronchioles, and we will talk about the alveoli in a few moments' time. One term that I do want to highlight here, and we'll talk about this term a little bit more in a few moments' time, but I do want to put it on the map already at this point. Let's see, how do I get, I didn't need to change the color of these. If I get this, is the term hilum. Hilum is the point where the primary bronchi enter to the lungs, and this spot where this entry takes place is known as the hilum. And just for the sake of, because my colors there jumped around a little bit, I'm just going to add the correct color here for the tracheotomy, and how I spoke about the air passing in. There was a blockage here. Okay, one thing that I do want to show as we are looking at the respiratory tree as a whole, we remember that we had our trachea, then our primary bronchi, our secondary bronchi, possibly tertiary, and then bronchioles, eventually terminal bronchioles, that then led to the alveoli. Is that as we are moving down this respiratory tree, we notice that the amount of cartilage decreases and amount of smooth muscle increases further down the respiratory tree we move. Next, couple of words about the lungs. Let's discuss about the lungs next. And for lungs, I'm gonna use this color. Lungs fill the entire thoracic cavity with the exception of the region of what we know as mediastinum. So mediastinum was, of course, the space where we find, for example, the heart and the great cardiac vessels. So it would be this space in between the lungs. Couple of terms that we want to notice there. I want to especially highlight a few of these. Let's start with the apex. So you remember the cardiac apex was the tip of the heart pointing to the downward, so inferiorly to the anteriorly, so to the front and to the left. Lungs have apex as well, but this time the apex of the lungs is at the very top, most superior points of the lungs. One on left, one on right. The base of the lungs is the inferior surface that rests on the diaphragm. And the term hilum we have already seen as the point where we are entering to the lungs with the primary bronchi. So those would be some of the very good ones to be familiar. Let's continue our discussion about the lungs, and I want to highlight a couple of things there. So we remember that the right lung is going to be larger than the left lung. And let's think of the explanation for the small size of the left lung, remembering that this was where the heart was located. So it takes a large portion of the left heart's or left lung's space. And actually we can see the indentation of the heart left to the left lung. And this is known as a cardiac notch. So it almost looks like part of the left lung is cut away or taken by this heart. And that is very correct analysis of what's happening there. What I'm going to do, I'm going to go in a little bit of a reverse order. So first, let's look at fissures of the on the surfaces of the lungs. So fissures were whenever we had this dip or a deep depression on a surface of something. We learned about that, for example, when we talked about the brain surface anatomy, when we talked about the cardiac surface anatomy. And we'll notice that on the right lung, we have a horizontal fissure and we have an oblique fissure. And these give rise to three separate portions that we will end up noticing. And let's mark those there. So we will have our superior lobe, we will have our middle lobe, and we will have our inferior lobe. And then I'm just going to erase those and simply write it instead. I think that's going to be a little better. So our superior, middle, and inferior lobe. On the left-hand side, we only have an oblique fissure that gives us a superior and inferior lobe to be aware of. So if you wish, that's kind of a nice division of how generally in most lungs we see the lobes and fissures. One thing to notice, of course, we have a lot of individual variation on that. So rarely would I find this to be the case in all of the lungs. We do get some variation. Lungs are surrounded, and again, let's do a quick sketch. They're surrounded by pleura. So here is our lung, the right lung, our left lung with the cardiac notch, our trachea coming down, primary bronchi. And all this is surrounded by our pleura. And pleura is made of two layers, the more inner one being visceral and more outer one being parietal layer. And in between these two layers, we end up finding fluid. And this fluid plays threefold purpose. First of all, it lubricates these surfaces so they slide past each other's nicely. But in addition to that, it also cushions a little bit of the lungs. But what's most important for us is the negative pressure that it generates within this space. And this negative pressure is really what helps us to keep these lungs expanded, as the lungs are continuously pulled to extend to their maximum size thanks to this pleural layer. If we have a puncture there, then somewhere here, let's add it on our diagram. Let's say we have a puncture here. Well, we lose the negative pressure and the lung will now collapse. And what we end up seeing is the clinical situation of pneumothorax. So that's what I wanted to say about pleura. Let's move on to the next structure, which are going to be alveoli. And again, just making a quick sketch for us, our trachea, primary bronchi, our secondary bronchi, maybe tertiary bronchi. Let's add some of those. And bronchioles. And terminal bronchioles then as well. And all this respiratory tree ends up at the very end with the structures that we know as alveoli. So what alveoli are, they are these very terminal parts of this respiratory tree, small kind of rounded structures there. And if we have a closer look of some alveoli, let's consider it as a little bit of a microscopic view. They end up looking like these sacs of thin-walled terminal structures of the respiratory tree. And what we notice is that the alveoli, next to them, we have a lot of blood capillaries running. And this is going to be for the pulmonary circulations capillaries. And what these capillaries do, they run very close of this very thin-walled alveoli. And this is what allows, through the process of diffusion, for the gases to be exchanged to the blood and to the lungs between those. So that's the important role of these that we want to be aware of here. The side of the gas exchange. And I'm adding our capillaries as well and highlighting them there. Okie dokie. That covers about the anatomy of the respiratory system, and next we are ready to move on to have a look of little bit more of a physiology side of the things. So we're gonna start by discussing the 4 phases of the respiration. And again, let me highlight. And the first phase is typically described as the pulmonary ventilation. So what pulmonary ventilation describes is really simply bringing in the air from the outside world to the lungs and all the way to the alveoli. And a couple of things to note here about the pulmonary ventilation is that breathing in is going to be an active process. So you need to actually make a conscious effort to get air entering into our body. So let's do a quick diagram again. Our trachea, primary bronchi, and our lungs here. And there's a couple of muscles that are involved in the process of breathing in. First of all, we have muscles in between the ribs, so intercostal muscles, and it's going to be the most external branch or external ones of those. And we also have diaphragm. This is the thin sheet-like muscle that we see separating the thoracic and abdominal cavity. Let's add that. Here. And it is the diaphragm that is the main respiratory muscle that I wanted to discuss a little bit about with you. What we see is that as you breathe in, the diaphragm becomes more flat. It goes downwards. And as that happens, our lungs in turn expand. At the same time, the external intercostals pull the ribs apart and our lung sizes grow. And as that happens now, we end up generating a low pressure inside the lungs, whereas outside the lungs, we've got a high pressure. And we remember the movement of the air is going to be dependent on the pressure difference. So air is going to move from the areas of high to the areas of low. So air is going to flow in as we do this inhalation, making the lungs bigger by recruiting our external intercostals and diaphragms. That's why it is going to be an active process. Breathing out instead is gonna be, so expiration is gonna be something that we can consider as a passive process that does not require a specific energy investment from us. If you think of taking a deep breath and you hold it, and once you relax, air will flow out naturally. That's because there is the elastic recoil of the chest cavity of the, especially our ribs, and the surface tension plays a role there as well. Of course, we can do forced expiration, which would be active. Here, we would recruit further muscles to assist us in breathing out and breathing out further than what we would normally do. This would involve muscles such as the oblique abdominal, transverse abdominal, and internal intercostal muscles. And as a result, whether it is traditional expiration or it is forced expiration. Let's have a look what happens there. Another quick sketch of a diagram. There are trachea, primary bronchi, our lungs. And now we are recruiting a bunch of muscles to assist us with breathing out. So we're recruiting our oblique abdominal, transverse abdominal, and internal intercostal. What all of these muscles help us to do is that they help us to make the chest cavity and consequently the lungs smaller. And as these are smaller, now we are generating a high pressure inside the lungs, whereas in comparison, it's going to be low pressure in an outside environment. And this is going to be the explanation for the direction of the movement of the air. So air always moves from areas of high pressure to areas of low pressure. So we are ending up breathing out by making the space of the lungs smaller. Okay, that was a rather lengthy explanation of the pulmonary ventilation, what we needed to know there. But we're going to continue to the next step, which is going to be about the external gas exchange. And that gives us a clue that there is probably going to be another kind, internal one, rather than just external. But let's focus on our external gas exchange at this point. And external gas exchange refers to this point where we are exchanging the gases from the alveoli to the capillaries of the pulmonary system. And also the other way around, from the capillaries of the pulmonary system to the alveoli. And this all was driven by the difference in the partial pressures at various different areas. So just the concept that we just talked about, that the air moves from an area of high concentration or high pressure to the area of low pressure. So breathing in and breathing out is explained with this. The next step that I want to discuss in terms of our phases of respiration is going to be the gas transport. So we move the gases within the systemic circulation during this stage. So the heart and the rest of the circulatory system there plays key role here. And now we get to the last stage, which is going to be, not probably hugely surprisingly, our internal gas exchange. So again, gas exchange, but this time it's going to be the internal one. So this is what's going to happen within the systemic circulation between the capillaries of the systemic circulation and either the interstitial fluid or the cells in there. So what happens now is that we have the oxygen moving to the tissues, and tissues give up the carbon dioxide to the blood within the capillaries. So that's our internal gas exchange. And of course, this process could proceed in the order that we just discussed, but also the other way around from the tissues or interstitial fluid all the way to the alveoli. So next thing I want to talk about, I've listed here bunch of non-respiratory air movements such as coughing, sneezing, crying, laughing, hiccups, even yawning. These are all processes where air moves, but it's not really to do with the respiratory process as such. And let's continue our review of the physiology of the respiratory system. Couple of terms that I want to introduce to you. Surfactant was a lipid protein complex that reduces the surface tension but also prevents the alveoli from collapsing, whereas I think of the term compliance as a measure of the stretch that the lung has. Next thing that I want to discuss with you is all to do with how do we assess ventilation and what goes on there. So you might remember that we had this tool, spirometer. We saw this plasticky thing where you breathe in, breathe out, and measures moved up there. These can be more advanced with computerized measuring as well. But the basic concept is very same. So with spirometer, we're able to measure the volumes of the respiratory system. And let's have a look at various different kinds of volumes that we can measure here. And we're going to start with the tidal volume, which is often abbreviated simply as TV. Tidal volume tells us about the amount of air that gets moved during the normal breathing in and out with each normal breath. The next volume that I want to discuss in respiratory system is going to be the IRV, which stands for inspiratory reserve volume. So this is the maximum amount of air that we can breathe in, inspire with extra energy. So in addition, beyond to the tidal volume. After that, the next one that I want to discuss is going to be the ERV, expiratory reserve volume. And this is the opposite of what we just saw, the amount of air that can be forcefully expelled with extra effort and beyond going further than the traditional tidal volume. And this leaves us with the residual volume, which is the measure of the amount of air that always remains in the lungs, that cannot be breathed in, cannot be breathed out. There's always some amount of air, for example, in the respiratory tree, or if we have alveoli that are non-functional, the air stays there but doesn't really participate in any kind of movement anywhere. Well, it was important for us to talk about the respiratory volumes because by knowing the respiratory volumes, we can then calculate respiratory capacities. And let's talk about the respiratory capacities a little bit and see how we can work them out based on the respiratory volumes. So the first one that I want to discuss about is going to be our inspiratory capacity. How do we calculate our IC, in other words, inspiratory capacity? Well, inspiratory capacity is calculated by adding together our tidal volume and our IRV, so inspiratory reserve volume. The next one that I want to discuss with you guys is going to be the functional residual capacity. So let's add that, abbreviated as FRC. And functional residual capacity, that can be calculated by adding together our residual volume and our expiratory reserve volume. The next one to discuss is then gonna be our vital capacity, and vital capacity, abbreviated as VC, is a measure that we can work out by adding together three: the tidal volume, the inspiratory reserve volume, and the expiratory reserve volume. Those three together. Well, the last volume that I want to discuss with you guys, and let's find a good color for that. We're going to use this color. Is going to be the total lung capacity. Let's have a look of that. Total lung capacity is calculated by adding together our tidal volume, our inspiratory reserve volume, our expiratory reserve volume, and our residual volume. So all of those volumes that we measured added together gives us the total lung capacity, as we see here. So this would be something that's useful to know as we are working out the different capacities based on the various volumes that we were able to measure with a spirometer. Now, I already mentioned a little bit referring to the concept of dead space within the respiratory tree, whether it's anatomical or alveolar, or the total, which is the summation of these two. In terms of the air that really doesn't contribute to the respiratory volumes or capacities, it just remains there. Whether it's because we're remaining within the passageway as an anatomical dead space, or whether it is because it's at the non-functional alveoli or non-functional parts of the respiratory system. But that's what gives us the dead space. I'm going to talk a little bit more about the gas exchange, and we talked about the external gas exchange and internal gas exchange earlier on. We're going to start this by looking at the external gas exchange, which is of course between the lungs and the capillaries of the pulmonary system. And what we're going to do here, we're going to look at how these gases are exchanged based on their pressure gradients, differences in pressure gradients. And we're going to look first at what happens with the oxygen. So what we'll notice, the blood that returns from the systemic circulation is going to have about 40 millimeters of mercury for the pressure of the oxygen, partial pressure of the oxygen, whereas the partial pressure of the oxygen within the air around us is about 100 millimeters of mercury. What this means is that we remember the movement was thanks to the partial pressure differences from areas of high concentration to areas of low concentration. So oxygen is going to be moving from the alveoli of the lungs to the pulmonary system's capillaries. Let's look at the movement of the carbon dioxide then next. Well, the blood returning from the systemic circulation to the lungs is going to have a partial pressure of carbon dioxide of about 45 millimeters per mercury, whereas the partial pressure of carbon dioxide in the air around us is going to be about 40 millimeters of mercury. So as a result, we move from area of high pressure to the area of low pressure. So the carbon dioxide will be moving from the capillaries of the pulmonary circulation to the alveoli of the lungs. That's our external respiration. Kind of a nice explanation of what happens there as a reminder, but also adding in a little bit more of this information about the concept of the partial pressures to there. Let's have a look next the concept of internal respiration. And we're going to look at the movement of these same gases with the basis of the partial pressure difference. And here we're looking at the movement between the capillaries of the systemic circulation and the tissues or interstitial fluid. It can be either one as it relates to the systemic circuit issues. And let's start again by looking at the partial pressures of oxygen. So now that we've exchanged the gases at the pulmonary circulation, the partial pressure concentration of oxygen that arrives to the systemic circulation is going to be the same 100 millimeters of mercury, whereas within the tissues, it's going to be 40 millimeters of mercury. So we remember again that the exchange was from the areas of high concentration to the areas of low concentration until the concentrations were equal. So as a result, oxygen moves from the capillaries of the systemic circulation to the cells or interstitial fluid. Let's have a look at the movement of the carbon dioxide. Carbon dioxide concentration in the blood that arrives to the systemic circulation is going to be about 40 millimeters of mercury, so same as in the airs around us, whereas within the tissues, within the interstitial fluid, it's going to be about 45 millimeters of mercury, so higher. So we remember the movement was from areas of high concentration to the areas of low concentration, which means in this case that the carbon dioxide will be moving from the tissues or from the interstitial fluid to the capillaries of the systemic circulation. And that review provided us a description of the internal gas exchange. So that's been a nice review of some of the basic concepts. And here we see, I didn't notice that earlier on, but explanation was, of course, this partial pressure differences. And we symbolize the partial pressure differences in writing by having the big P, and then it can be either oxygen or partial pressure of carbon dioxide in here. So depending on which one we're looking on these diagrams that we just discussed about. Okay, let's have a look of how these gases, oxygen and carbon dioxide, are transported within the blood. That's going to be something interesting to consider. Well, oxygen is mainly transported bound to hemoglobin. That's going to be about 98% of oxygen binds to the hemoglobin. You remember the 4 heme groups in a single red blood cell, right? That allowed one oxygen to tie into each of these heme groups. So in total, a single red blood cell was able to transport about 4 oxygen molecules at the maximum. The remaining 2% or so is transported bound into the plasma. Let's have a look at the carbon dioxide transportation. Well, about 3/4 of the carbon dioxide is transported as bicarbonate, remaining 15% as hemoglobin and 10% as bound to the plasma. So that gives us an idea of those. The clinical relevance that I've highlighted here, or that would be nice to know, is the concept of hyperventilation. You remember in hyperventilation, the patient is breathing out too much carbon dioxide. And that will become an issue unless we're able to stop that vicious cycle. Some of the old school solutions to that have been breathing into a paper bag, which then catches up some of that carbon dioxide that's expelled so that it doesn't all escape the body. We breathe that back in from that paper bag, that concept. That really covers everything that I had to say about the respiratory system. And the next system that we'll have a look of is gonna be the urinary system. And we're gonna go through all parts of the urinary system, but we're gonna focus especially on the nephron as the key structural and functional component of the urinary system. Kidneys are the first ones that we're gonna start with. So we're going to mention a couple of words in terms of the kidneys. The main function of the kidneys, as I've highlighted, has all to do with the blood pressure regulation, also regulating the total fluid, water volumes, solute concentrations, acid-base balance, and so on. But really the big one that I wanted you to remember was the blood pressure regulation through the kidneys. Of course, we also excrete, get rid of some of the metabolic waste, toxins, drugs, so filter the blood for that as well. Let's have a look at some of the external anatomy of the kidneys. And kidneys are often described as these reddish-brown, bean-shaped structures that we have. And we're going to look at the external anatomy of the kidneys to start with. And there's a couple of characteristics that I really want to highlight there. So we notice that medially, the kidneys are concave. So there's a smaller curvature that faces medially. And we see various different vessels coming into this side, the renal hilum side. We have our blood vessels, both arteries and veins coming, arteries in and veins out, and that tells us that we have a very rich blood supply to the kidney, which tells us that, of course, the blood is coming there to get filtered and then leaves. In addition to that, we have nerves coming into the kidney, and we even have lymphatic vessels leaving the kidney as well. So a lot of stuff coming in, a lot of stuff leaving, telling us about the importance of it. One thing that we'll notice there is that we also have a ureter leaving the kidney. So the urine that has been produced gets picked up and then transported to the bladder by the ureter. So we'll have a look at that a little more in just a moment. The lateral side of the kidney, if we think of the external anatomy, we'll notice that that is convex, so larger curvature there. And we'll also notice that the surface of the kidneys typically appears as smooth, shiny, and that's because it is enclosed inside a capsule known as a renal capsule. If you want to discuss about the location of the kidneys, where are the kidneys located? They are retroperitoneal, so in terms of the peritoneum, they're behind it. Let's have a look of a little bit of the location of the kidneys. So let's circle them here. We end up finding that the right kidney is located slightly lower than the left kidney. And you might be wondering, why is this the case? Well, the explanation for this is going to be the fact that we have right above, superior to the kidneys, the liver, and liver takes most of the space on the right side of the superior portion of the abdominal cavity. So that takes some of that space. And for that reason, the right kidney has to be a little lower. One more thing referring to the retroperitoneal nature of the kidneys, do notice that kidneys are very far back. So closer to the back of your body than most students end up thinking. Let's have a look at the internal anatomy of the kidneys a little bit. And I'm going to sketch a diagram here to help us to discuss that a little bit. Let's see if we can do that nicely here. So we're going to draw a kidney with a cross section of it. Here we have our ureter coming out from there, our major and calyx, and we also end up seeing some renal pyramids. And we're going to color all of these and make sense of all of these structures. I just prefer to sketch them ahead so I remember to tell you everything you need to know. So let's have a look of the internal anatomy of the kidneys. So kidneys do have a renal cortex, and you remember the cortex, we talked about that also in the nervous system, for example. So that's the most superficial layer. So renal cortex is from the inside of the kidneys the most superficial layer that we can see there. In addition to that, we, of course, have our medulla, which is the deeper portion of the internal structure of the kidneys. So medulla would cover the rest here. And within the medulla, we can see certain structures that I want to highlight. Let's highlight a renal pyramid. sometimes known also as medullary pyramid. They are these triangular shaped structures within the medulla of the kidney. I think I drew quite a few of them to this diagram, so bear with me as I color them in so that it makes sense that what they are. And we also have the area of the renal pelvis. So this is the medial funnel-shaped part. We see there a calyx that collects the urine produced and then funnel it to the pelvis. And from pelvis, this urine then leaves through the ureter towards the urinary bladder. And I've listed there also the flow of the materials through the kidney, starting from the renal pyramids and ending to the ureter. Let's have a look at the nephron. I mentioned earlier on that the nephron is the most important structure that we'll be looking on the urinary system. It is the structural and functional unit of the kidneys. So let's go through that. And how we're going to go through that is by looking at the different parts that you should know of the nephron. And just as a disclaimer, and I'm only sharing this with you because I had to clarify this to one of my students in the previous, not even in this school where we're studying now, in a previous school where I was teaching this student, because I often used a diagram of a nephron showing the nephron one way around. Argued that the structures have to be named, that they always start from the left to the right. Of course, nephron can be shown in any direction. I just, for the sake of simplicity, try to follow the same pattern whenever I draw a diagram to you, but it could be shown in any direction. So let's have a look of some of the structures that we want to know about the nephron. First of all, let's have a look of the blood circulation. The blood vessel coming into the nephron here, that's going to be our afferent. Arteriole. And the blood vessel that leaves the nephron, that's going to be our efferent arteriole. We also end up seeing the glomerulus, and glomerulus is this capillary network within the nephron. And I'm just, for the sake of completedness, also gonna add this capillary that goes around from the glomerulus all the way following the nephron in a close distance. And we'll see the purpose of that in terms of physiology in just a moment. But I'm gonna label our glomerulus here. Okay, what other parts of the nephron might we want to know? Let's start labeling the actual nephron itself. We're going to start with the glomerular capsule, which goes around the glomerulus here. And I'm trying not to color over the other structures that we've either seen or we will see. But that's going to be our glomerular capsule, or sometimes known as Bowman's capsule. Then we have our proximal convoluted tubule. That's the part that's closer to the glomerular capsule of the two convoluted tubules. And then we have, of course, our distal convoluted tubule as well. Let's label that as well. And in between of those, we have our nephron loop, which is sometimes also known as loop of Henle. And finally, the last part, which is going to be our collecting duct. Collecting duct receives the urine output from multiple nephrons. And then passes them towards the calyx. So that's our nephron, the main parts of the nephron that we need to know. And I'm going to add one more thing to this diagram. We're going to have a look of and consider the movements of the fluids. And let's start with the process of filtration. So filtration is when we are taking the contents of the blood from the glomerulus and passing it to the glomerular capsule or Bowman's capsule, the first part of the nephron. Let's add that here. The second movement of the fluids that I want to discuss with you guys is going to be the reabsorption. So in reabsorption, the fluids that were taken by the nephron from the glomerular capillary network are now put back into the blood circulation in a process of reabsorption. And next one that I want to add there is going to be the process of secretion. In secretion, we now are able to add some of the fluid content from the cardiovascular system, from the capillary that runs next to the nephron, back to the nephron in a process of secretion, as I said. And the final one that I want to map here and mark for us is gonna be the excretion. And excretion is basically just whatever gets put out from the nephron as the urine that leaves the nephron and then gets dumped into the collecting duct. If you wanted to, if you're mathematically inclined and find formulas very comforting and easy for you, you can consider that the excretion is the filtration minus reabsorption plus secretion. So that can be considered as kind of a formula to explain the movements of these fluids within the nephron and physiology of the nephron. We're going to discuss a little bit about the concept of the glomerular filtration rate, often abbreviated as GFR. That's an important measure that you often see on the lab reports. And what glomerular filtration rate tells us, it tells us how well the kidneys are working, basically. So how much blood passes through the glomeruli in a single minute. And earlier on this course, we have talked about antidiuretic hormone, ADH. That might ring some bells from our endocrine system chapter. We remember what antidiuretic hormone did was that it was anti for pee production of the urine. So if we have a great amounts of antidiuretic hormone available around, that results in less urine being produced. And where antidiuretic hormone worked was at the distal convoluted tubule. So if there was a lot of ADH present, it made the distal convoluted tubule very permeable. A lot of the content of the nephron was then absorbed back to the body. Give me a second. I'm just going to go check. I live in a rural side and I hear a lot of coyotes outside. Just let me go and check that for my pet chicken, that everything's okay. I'll be right back. Okay, and we're back again. One of the joys of rural living is that you do get to deal with the wildlife coming close to the house as well. But I think that we've finished all that I wanted to say about kidneys and the nephron. We are ready to look very briefly of the ureters and urinary bladder and the urethra. And to discuss all of them, what I'm going to do, I'm going to draw a little sketch diagram for us again. So let's draw our kidneys with the left kidney a little bit higher than the right kidney. Then we have our ureters coming out of those nicely. Remember, they wrapped up and around the urinary bladder, entering in there on the base of the urinary bladder, actually. And then we have our urethra draining the urinary bladder. So let's go through those nicely and quickly. And we have talked about the kidneys before. So I'm just going to color them there for us to be able to follow the passage of the urine through the system. That's good there. And we are ready to move on to discuss about the ureters. So what ureters do, they run from the kidneys to the urinary bladder. They're about 25 centimeters long, smooth muscle tube. And the fact that they're smooth muscle is going to be important for us as we think of the fact that the urine moves along these tubes, even at the time when we are standing against the gravity or are in zero gravity are in zero gravity environments such as astronauts into space, the urine will still move from the kidneys to the urinary bladder. If you do want to consider again the relationship to the peritoneum, you remember kidneys were retroperitoneal, so are the ureters, so they're the behind the peritoneum. And I mentioned the importance of the smooth muscle wall of these tubes. And it is the movement of peristalsis that moves the urine within these. Next, I'm going to talk a little bit about the urinary bladder. If you really want to simplify things, you can say that urinary bladder is very little anything else but a temporary storage pouch for the urine. So it can stretch and shrink depending on the needs. Again, if we think of the location, it is going to be retroperitoneal, so behind the peritoneum, and located at the pelvic floor. We'll talk about the trigone in a few seconds. I do want to talk first, though, a little bit about, I didn't mean to do that. A little bit about the urethra. So urethra is a muscular tube that drains the urinary bladder and opens it to the external world. And what's important to notice there is that we've got a bunch of various different kinds of sphincters. Some of them, especially the more internal ones, are involuntary, whereas the external ones are under the voluntary control. But we'll notice the male urethra is much longer than that on the females, which results that the females with the short urethra are more likely to have bacteria traveling up to it, to the urinary bladder, and getting the urinary tract infections. So now that we have discussed about these main structures, one thing that I promised to come back to discuss with you guys was the concept of trigone in the urinary bladder. So what we're going to do, let's take a section across the urinary bladder here and have a look as if we were looking down to the urinary bladder from the top. And what we'll notice that at the base of the urinary bladder, there's going to be 3 openings there. And again, for the sake of completedness, let's color in our urinary bladder using the same color coding. So bear with me as I'll do that. So this is all as if we were looking down to the urinary bladder from the top. Very useful anatomical landmark if you end up doing, for example, imaging, if you end up doing, say, ultrasound, the trigone comes in useful landmark in that you know what you're looking, what you're seeing. So that's our urinary bladder. And the 3 openings that we see there. Let's list them. I'm going to, for the sake of completedness, just add a note that we are seeing at the bottom, the anterior side, and at the top, the posterior side, the back side. And the three openings are basically two of them, the more posterior ones, are going to be our ureters. And the more anterior one, that's going to be our urethra. And the reason why I wanted to discuss about this is that if you look at these, they form almost like this triangle shape, don't they? With these three openings. At least if you have a little bit of imagination, you can see kind of the triangle shape. This is known as a trigone. And trigone is, like I said, useful landmark when we're studying the structure of the urinary bladder and at the base floor of the urinary bladder. So that's the reason why I wanted to discuss that briefly with you. Okie dokie. One more thing that I want to mention about the urinary system, or a couple of more things. First of all, the proper word when we talk about urination in anatomy and physiology in medical field, that is known as micturition. And let's have a look at the composition of the urine. Majority of the composition of the urine should be water, about 95%. About 5% of it is various nitrogenous wastes such as urea, uric acid, creatine, and then others that I have listed there as well. But largely water. And let's have a look at what could be some of the problems with urine if we are talking about that. If we have too high concentration of any component that we normally find there, that could be a problem. And also, we do not want to find in the urine, we don't want to find blood in there. Not red blood cells, not white blood cells. White blood cells, you remember, was a sign of that there's an infection of some kind, not bile pigments. But really, from my perspective and my clinical background, I would really say that it is the red blood cells that we would be concerned if we see blood in general as well in the urine. So we don't want to see that in the urine. And I think that that completes everything that I had to say about the urinary system. I know that there was a little bit more brief review. Pay attention especially to the nephron. That's an important structure. I think we did a fairly good job of covering that. And final chapter that we have to look at is going to be the chapter that covered the fluids, the electrolytes, and the pH of the body and balance of all of those. So we'll spend the remaining time on our review of this study guide by looking at these three and their balance. Much of this chapter should be familiar revision to you, but I'll take a little bit of time to make sure that we are all on the same page and at the same place. The main purpose why we do need to know about this is that we need to know what's normal so we can understand what's abnormal. And for example, a lot of lab tests, you would need to be looking at typical values so you can figure out if the values of the patient fall within that. In your last week's critical thinking assignment, I think you were looking at a lot of these lab test values, what are normal, what are abnormal. So on that note, let's make a start and start by looking at the water content. So all living organisms, we end up noticing that they contain large amount of water. Same is true for humans as well, and this could vary from anywhere from the 45 to the 73 plus percent of the total volume of the human being of water. We'll notice that adult males have slightly more water content than adult females. That's because females do have a bit of higher fat content and less skeletal muscle mass on average. Whereas we noticed that the water content of the infants was especially high because of their low body fat concentration and low bone mass, whereas the elderly people, we can get as low water content as 45% of the volume. Well, where do we find this water then? Let's consider that a little bit. I'm going to draw just again as a visual aid for anyone who finds it useful. A diagram, here's a couple of cells that we see with their nucleus in the center. That's all that this sketch is showing. And we're going to be looking at where do we find water at different parts of the body. So about 2/3 of all the water in the body is located inside the cells. So that's gonna be intracellular fluid. So majority of the water in a human body located inside the cell. About 1/3 of the water in a human body instead is gonna be located outside the cells, known as extracellular fluid, or ECF, if you wish to use abbreviation. And places where we find this are, for example, within the interstitial fluid, but also within the blood plasma and the lymph. So again, some amount of the fluids are located also outside the cells, but at large that volume is lesser. You should have discussed on your Bio 181 or on your Bio 156, depending a little bit which course you took, the basic biology course regardless, about characteristics of the water. So we'll discuss about that very briefly. I want to mention 7 characteristics that are especially important. We know that water is amazing, excellent transport media. So it is used to carry a lot of things. In humans, a majority of the blood is made of water, so we know blood is a major carrier of gases, nutrients, waste products, and what else could I think of? Hormones, and so on and so on. Water, 2 characteristics that played a major role there in the behavior of the water is the cohesion, which was the attraction of the water molecules to other water molecules, as well as adhesion, which was the attraction of the water molecules to other polar substances or surfaces. In addition, we know that water holds a high surface tension. This is because of the cohesion and allowed, for example, many bugs to walk on the surface of the water and so on. Water also worked as an amazing as a heat reservoir, so binding heat and releasing it slowly. So it evens out, stabilizes the changes of the temperature, especially at the coastal region. That's why many people prefer to live by the big bodies of water, because you don't get too hot during the day, you don't get too cold during the night. It kind of works as a buffer stabilizing. We also know water takes three main forms that we have discussed: solid, liquid, and gas. There is a fourth form that can sometimes happen, the plasma, but we'll talk about that in further studies. And the final point, which is really important, is that water is an excellent solvent. So many things, many solutes are capable of getting dissolved into the water. So really, if we take these two characteristics, that water is able to dissolve a lot of things, and then water is able to transport these dissolved solutes in it, that makes water so ideal for our purposes in biology when we look at, for example, human body. But it doesn't just have to be the human body in terms of as a transport media. You can also think of the nutrients carried into water to the plants and so on and so on. Well, when we look at the characteristics of water, there is going to be a bunch of solutes that don't dissolve to the water, such as glucose, lipids, urea, and creatine. These are not so much of an interest to us. We are most importantly interested about the solutes, which when they dissolve to water, they end up with charges forming. And these charges are often indicated as such as here as either positive or negative that result from these things dissolving. And those affect the behavior of these materials. One big thing, if you learn nothing else from this chapter, what I would like you to know is that where water goes, the electrolytes will follow that. And similarly, where electrolytes go, of course, the water will follow them. So both water and electrolytes are connected to each other in terms of their behavior. So you can think of them as being interdependent. So dependent on each other. So that would be a good point to get out of this portion of this chapter. Let's have a look at what else I have highlighted here for us to take away from this chapter. I'm just going to zoom in a little more. I have discussed with you a little bit about the intake of water and output of water. And one big thing that we noticed, it was that these two should be in balance. So we should be taking in the same amount of water as we are losing every day. The intake of the water was driven by the thirst. So about 2/3 of all the water that you get in is from drinking alone. Also, some amount of water to the body enters through the moist food and also as a byproduct of the metabolic processes. So often there we end up producing water as a byproduct. In terms of getting rid of the water, that's mainly through the urination. Again, about 2/3 of all the water loss is through urination. Some amount also through the evaporation, sweating, and also being breathed out. Very small amount of water loss through the feces, because feces are relatively dry matter. Next thing that I've done here has been that I wanted to discuss with you guys about the concept of osmoreceptor ADH mechanism. And this is an important mechanism as we talk about the water balance in the body. And many textbooks give quite complex diagrams and descriptions of this. Me instead, you know me, I like to keep things simple. So we go back to our good old friend of the concept of homeostasis. And you remember that, of course, homeostasis was all about maintenance of consistent internal environment. So this homeostatic mechanism loop, which I provided a short description here or short visualization here, that can be used. Oh, I didn't mean to do that. That can be used to explain also this osmoreceptor ADH mechanism, which is a rather complex process, but we're able to cover it very simply by using this diagram. So that's why I was so big on let's learn the basics well so we can apply them as we need. So let's start by looking at a situation where we have too much water in the body. So you can think of this as if the blood, for example, would be too watery. So that's our stimulus, something that has changed. And how are we able to detect that? We're detecting that with the osmoreceptors, which are located at the hypothalamus within the brain. So that's our sensor. And of course, the osmoreceptors pass the message to the other parts of the brain where we compare that to the normal. What should be the normal amount of water in the body? How watery should the blood be, if you wish to use that expression? And if we're beyond this normal amount, then the effector kicks in. And the effector in this case would be our pituitary, which would secrete less ADH. And if we make less ADH, you remember ADH was our anti-urination hormone. So if there's a large amount of ADH, we make less pee. If there's now a small amount of ADH, we actually end up making a lot of pee, so more water is recovered from the distal convoluted tubule, and as a result, a lot of pee is being produced now, so we can pee away a lot of that extra water that we wanted to get rid of. And that's how our homeostatic mechanism loop is able to explain the osmoreceptor ADH mechanism as it relates to when we have too much water in the body. Let's look at the opposite situation where we have now too little water in the body. And let's see what we can learn about that, what sort of observations we can make here. So the starting situation is that now we have too little water. You could think of it almost as if the blood would be too salty, so there's not enough water on that. Again, where is this stimulus? The sensor is going to be the osmoreceptors within the hypothalamus, and they pass the message to the brain, which is our control center, where it gets compared to what the normal should be. Are we within the normal range, or are we off from the normal range? Well, if we are off from the normal range and we have too little water in the body, now the pituitary gland starts to secrete more ADH. And again, we remember that was the anti-urination hormone. So as a result of that, more water is taken back at the distal convoluted tubule, and we end up producing less pee, less urine is being made. So that's the concept of how the osmoreceptor ADH mechanism is able to conserve water when there's too little water present. So I think that these two diagrams are really simple, much more simple than many other versions that I've seen on textbooks for explaining this kind of a complex concept of osmoreceptor ADH mechanism. So trust me when I say that there's a reason why we learn also the complex topics or learn the basics well. That helps us to explain the complex topics. Couple of other things that I wanted to discuss with you. I want to discuss about the concept of tonicity a little bit. Let's start by looking at the situation of hypotonicity. What happens in hypotonicity? So hypotonicity refers to a situation where the insides of a cell are more salty than the outsides, which outsides of the cell are then more dilute in comparison to the inside of the cell. As a result of this, now water will be flowing into the cell, and as water flows into the cell ends up swelling, so growing in size, and it might eventually even end up bursting. So that would not be ideal. That would be a little bit of a problematic for us. So this is how you can kill yourself by drinking too much water, for example. So how do you remember that hypotonicity refer to this? I always say try to make up mental rules, mental visualizations of any complex concepts, and it will help you. So what I'm trying to do here, and I know I'm not super successful, I'm trying to draw a diagram of a hippo. It looks more like a pig and a cross between pig and maybe koala. But that's the best I can do. So when I hear hypotonic cells, I think of hippo cells. Hippos are these large, swollen animals, and that large swelling describes these hypotonic cells, what we're seeing happening there. Let's move on to have a look of the isotonic cells. What happens in isotonic cells and what we should know about them. So in case of isotonic cells, what's going on is that the cell, both inside and outside the cell, have the same amount of salts. So the concentrations are the same both inside and outside. That bothered me. So as a result of that, as some water will always enter the cell, the same amount of water also leaves the cell. So the overall amount of the water within the cell will remain the same. So there's a zero net gain or loss of water in this case. And how I think of this is that if I see isotonic cell, I think of those isotonic cells say that I so perfect. They're perfect, they don't need to change, they can remain the same size. And the final tonicity case that I wanted to discuss with you guys is gonna be the concept of hypertonicity. Let's have a look of what's going on hypertonicity. In hypertonic cells, the inside of the cell is very diluted, whereas the outside of the cell is more salty in comparison to the inside. As a result of this, water is leaving the cell and flowing to the outside. So the cell will end up shrinking and it might eventually even end up drying up. Well, how do I remember this? I think of what kind of are those people who are very hyper. Well, what happens with the hyper people that I know? They are very active. They do a lot of things. They're constantly going at it. And if you're very active, what happens? You become also very skinny. So my mnemonic for hypertonic cells are that they're hyper cells, they're my skinny cells, and they end up becoming very skinny, shrinking as a result of this. So there you have it, my mnemonics for different tonicities. I hope that they help you. I find them helpful, but always make up your own rules if you can think of something better. And I'm always curious to hear about those too. How this all relates to what we have discussed. Well, this all relates as we think of what happens to the electrolytes as you, for example, exercise a lot, you need to replace them. Just simply replacing the water is not enough. And certain components of these recovery drinks also help with enhanced water uptake. So that's why this matters. Where the fluids go, where the water goes, the electrolytes follow, or the salts follow, as we saw on this topic that I said. If nothing else, do remember that the water and electrolytes are interdependent on each other. Okay, that moves us to the last larger topic of this chapter. And I'm going to try to find the right color to talk a little bit with you just briefly about the pH scale. A lot of this will be familiar and it will be revision. Hopefully we can add a little bit more substance to that as well. So you remember the pH scale ran from 0 to 14 with the pH of 7 being the neutral. And if we had a pH that was above 7, we talked about that as being alkaline. Or, in other words, basic. If it was below 7, that was acidic. Well, I think that is all familiar to us. Most of us in our discussion. What I do want you to remember about the pH scale is simply a mathematical representation about the concentration of hydrogen ions in that substance. So it just simply tells the relationship, how much hydrogen ions we have somewhere. And what we end up finding is that in the body, the body maintains for example, the blood pH within a very narrow range between 7.35 and 7.45. So there's very little variation in our body pH, and it doesn't matter what you eat. If you eat alkaline diet or whatever, your body pH will remain the same. What might happen is that you eat certain kinds of foods that are just better for you because they're healthy foods because of how you think of them, but you are not going to change your body pH by eating something, not significantly, if you're a healthy and normal human being. What happens is that this body pH is kept within such a narrow range, and even the survival range to me seems to be very narrow, between 6.8 and 8. If you go anywhere below that or anywhere above that, you will die. That's a narrow range, so that's quite an accomplishment for our body to be able to maintain the pH within such a narrow range. So let's have a look at how this is possible. There's 3 ways how we can do this, and these 3 can all work and play a role. So first of all, we have certain chemicals that bind either to acids or bases, and in this way neutralize them. This is a very fast way of dealing with changes in the potential changes, threatening changes in the body pH. That's our buffers. The second way how we can deal with this is through respiration. This is a little slower, takes about couple of minutes to act, and that's all about breathing out what we don't want to have in the body to neutralize the pH balance of those changes. And the final method that I'm going to discuss with you just briefly is the renal method. It is slowest, so it can take anywhere between hours and days to get rid of materials via kidneys, such as hydrogen ions, getting rid of them via kidneys in urine. But it is also the most potent, so it's very, very efficient way of balancing of the pH and bringing the body pH to the normal range. So these are the 3 regulation methods of how we maintain our body pH within such a narrow range. And the relevance of this, the clinical relevance would be that if we have too high of a pH or too low of a pH, we have then some clinical consequences with this. We can talk about alkalosis or acidosis, and I provided quite a good description of some of the signs and symptoms in our last week's lecture video and revision as well, so you can refer back to that. The very last concept that I want to mention with you guys is the concept of the fluid therapy. So basically putting an IV drip on a patient. This is often one of the very first things that we do when a patient comes in, and that's simply to help to maintain the blood volume, but it's also because it is a great route of quickly introducing certain medications to the patient, to their bloodstream, without it taking a long time. So that's why fluid therapy, putting an intravenous IV drip, is one of the very first things that often gets done in the hospital. What is that IV fluid then that we give you? This is basically, if you want to be really mean, you could say that it is expensive salt water. And what I mean by this is that this fluid, these liquids have to be sterile, and they have to contain just the right proportion of various different electrolytes for optimal functioning of the body. So it is more than a salt water. It is a carefully balanced, it is sterile, but it really is nothing that much more amazing or secret, as you would have probably thought, than just your typical fluids. But they're fluids where you have just the right amount of these electrolytes for optimal functioning of the human body. And on that note, that completes our review of the chapter materials for this exam. So we have covered the chapter on the respiratory system, the chapter on the urinary system, and the chapter on the water electrolyte and pH balance. So I think that with that, we've done a pretty good job of covering everything that I wanted to cover here. So I'm going to jump off from this presentation, and bear with me as we jump back into the PowerPoint that we were looking at earlier on. And on this PowerPoint, I just want to wrap up what we're talking on this session. I want to remind you that we have a rather nice list of terms that you need to know also in terms of the lab practicals. So those are available on your Canvas learning module for this week. And I strongly recommend that you complete the lab practical practice. You might even want to do it a couple of times so you get a chance to see all of the possible questions from that question pool that might come up in your actual practical. So that, remember, these both the exam and lab practical are open book, so you can take all your notes out. You can have anything that helps you. You can take pictures, screenshots of the practice versions if that assists you, and just make it easy for yourself. Give yourself a chance to succeed to your very best ability, and complete that. Remember that they are timed, these exams and practicals. Unless you have an accommodation plan, then you might have certain changes from that typical timing that we would normally have. Quick shout out, the lecture slides, someone was asking for them. They are available from all of these chapters that we've covered. They're not the exact same slides that I use in my presentations for copyright reasons, but they are the slides from your textbook provider. And as I've said earlier on, I am very happy to open or extend any assignments that you have. All you have to do is message me with quite a bit of clear details of what assignment it would be, and I will definitely work with you. I want you to get those points. You do have tutoring available, even though we are still working remotely. You do not need to have an appointment. You simply log into the Cranium Cafe and find a tutor that works with for you, that you find that could assist you, and then you work away. Remember that I'm also happy to help you. I've done a lot of individual sessions recently. All you have to do is reach out to me, and I'll set a time with you when we are both available, and we can go through whether it is technical matters of your course, whether it is content related, or whether it's something else. I am here to assist you and make sure that you can be as successful as possible on this course. Any questions, feel free to contact me. I am really here to assist you. I want you to succeed. And like I said, I know that I've got a couple of messages, and I'll get back to those immediately as soon as I'm done with this. And on that note, that really covers everything that I had set out on my mind for a goal for us to do on this review session. And on that note, I thank you for joining me, and I really hope that you have a very good week. Try to get that exam and lab practical done. If you have any work to catch up on, this is a good week to do that. I don't want you to have to hustle on the last week of the course. You can do it. I thank you. I hope you have a very good rest of the week. Remember, I'm here only a message away, and I'll help you. I'll see you either on Wednesday sessions or I'll see you next week. Thank you.